Yeah, I'm gonna give this to you.
All right, I will talk briefly here. So I believe the only reason that I'm getting to give the introduction is because I'm the other person who owns furniture in this department. So it is a great pleasure to have Peter Greer here in this capacity as the new Stauffer chair in cancer research here at Queen. So Dr. Greer originally graduated from McGill University with his BSc and went on to do his PhD there with Stuart Nord, correct? He then went on from there to the Lunenfeld Institute in Toronto, where he was part of the illustrious stable of researchers who came out of the Pawson lab, all of whom I think essentially have done amazing things for cancer research here in Canada and internationally, not the least of which is Dr. Greer. He's continued on since then, as he said of another speaker in this position, to hold almost all of the alphabet's worth of research funding from institutions across Canada. He's owned MRC, CIHR, NCIC. Some of these are institutions that you younger people will never even heard of, but Canadian Cancer Society, Cancer Research Society, OICR, TFRI. He has had his finger in almost all of the pots of cancer research funding here in Canada. He has been the groundbreaker in research on FAS and fair oncogenes, and I won't mention more about that because he's going to tell us about that today. But he's also had a strong collaborative interest in calpain and in the Ezrin Moesin family of proteins as well. I think some of the things that don't show up in Greer's CV, but are really important, is that he's had a hand in the training of essentially every graduate student who has come through our department over the last 20 years. If he hasn't set their committees, he's introduced their seminars, and he's organized scholarship applications and successes for all of them. And so I think we as a department and our collective students really owe Dr. Greer an immense vote of thanks for his contributions, as the mandate of the chair says, in terms of educating and mentoring the next generation of cancer researchers. So I think we should welcome Dr. Greer. And what I should also note is the first time in 30 years he's ever actually given a talk in this department. So Dr. Greer, if you would.
And you're about to find out why I don't give talks. I'm good at telling other people how to give talks, but we'll see how good I am at it in a minute. So thank you very much for those very kind introductions. And you covered some of the things that I was actually going to start with. So I'm going to tell you a little bit about FES, and this is kind of a long story. And I've tried to pick out little vignettes that will give you a bit of a taste of it. So I do want to thank Roger Deely. So this guy recruited me when Paul was chair, actually. Well, both of you recruited me to this department because pathology was my home when I came here. And Roger was a mentor for me, helped me to establish my research program, helped me read my grant applications in those first years. And we know of Roger and Susan's important groundbreaking work in discovery of multi-drug resistance proteins. Susan Cole and Roger Deely published this in Science. And that really put them on the map internationally because this was a huge discovery at the time. But Roger also did lots of other things locally, provincially, and in Canada when it comes to leadership in the cancer research community, including the establishment of the Cancer Research Institute, now the Sinclair Cancer Research Institute. So I just want to thank Roger, and I feel a little bit of that imposter syndrome actually taking on this chair position that Roger first had. I also want to thank this gentleman here. So this is my postdoctoral mentor, Tony Pawson, who passed away 11 years ago. And Tony was, as the other Tony, as they call him, Tony Hunter. Tony made important discoveries in how signal transduction works. He described some of the first protein modules that were involved in and allowing different signaling proteins to talk to one another. And I think his work will eventually have a huge impact. Everything in cancer seems to be incremental, but I think that Tony's work, if he had survived, probably would have earned him a Nobel Prize. And I think it will have huge implications, and already has, in how we target different aberrant signaling pathways in cancer. So I've been very fortunate to be able to recruit a huge number of very excellent students, postdocs that have come through my lab over the years. And so I want to thank you guys. Some of you are in the room here today. You inspire me. And as I always say to you, when you leave this lab, and probably well before, you'll know more about this stuff than I do. And so that's enabled me to keep learning all these years by interacting with these people. But it's also just a lot of fun. So I thank you for inspiring me to come to work every day. I've had quite a few trainees come through the lab over the years. I've tried to make a complete list of them here, and also a number of staff, technicians, research associates that are all listed here. But I've also been very blessed by finding a lot of people, both locally and nationally and internationally, to collaborate with on the various projects. So as Lois mentioned, FES and FAIR have been mainstays. And so I started working on FES and FAIR tyrosine kinases as a postdoc in Tony's lab. When I came here quite a few years ago, 25 years ago, John Else got me interested in calpain, and that turned into a very big program. And two of my graduate students sitting here are PhD students working on the calpain project. And then Bruce Elliott, I think, is somewhere here. There he is. Bruce, thank you for introducing me to Ezrin. So that's turned into a very interesting project. And I think all of these have potential to be therapeutic targets in cancer, and we're trying to develop inhibitors for all of them. And as Lois mentioned, I've been really lucky when it comes to funding from various organizations. Queen started by giving me startup money, and so that was a great help at the outset. But I was lucky enough to get my first two grants from MRC back then, which became the CIHR, and from the NCIC and Canadian Cancer Society. And I've been lucky enough to get funding from all of these other organizations. So now I guess I get to add this one to the list, and I'm very grateful for the Stauffer chair. So one other thing that gets me excited about all of this is my family. So thank you, Jen, James, and Jordan, for coming here tonight. I was very blessed in having not just an opportunity to build a career here in Kingston, but also to start a family. So I met Jen, and these are our two kids. This is a very old picture, but it's a favorite picture of mine. My brother Tom took it. So you can see, I don't know how old they are here, probably 4 and 6 or something like that. But it's 20 years later, and they've grown into two beautiful adults. And so now the story. So this story starts sometime in the 70s. What was I doing in the 70s? I was a high school student at Westmount High School. And I swear this is not me. It's a guy in my homeroom, and for some reason I can't remember who he is, because that's usually the posture that he had at homeroom in the morning. This is me. Back then, long hair was a thing. You'll see I had a lot more of it at this point. And my dad was a major in the Army, and until about 5-6 years before this picture, I was sporting a crew cut. But he retired his military issue clippers about 5 or 6 years before this. And as I say, back in the 70s, long hair was a thing. There were a number of really important graduates from this high school over the years. This guy graduated from Westmount High School too. And so did this person. And hopefully she's piling up lots of votes today so that tomorrow we can all be singing Hallelujah together. And you'll notice that she has a shout out to her mother in this yearbook entry. And some of you may also know that she was a cancer researcher at McGill University. And she studied estrogen signaling in breast cancer. And I believe she died of breast cancer. So Kamala Harris, good luck today. Was it cancer? Okay, I knew it was cancer. But, so what were cancer biologists doing in this period of time? Well, one of the things that a lot of them were doing was looking at retroviruses that had been isolated from tumors that arose in chickens and cats and other animals. And what they noticed around this time, we could actually look at genomic sequences. And what they found was that there was some mystery sequence in a lot of these viruses. And I'll just get my pointer up here. Oops, not my laser pointer there. Yeah, there it is. There was very often this foreign sequence in here that was called a V-onc. And that was because these were the sequences that were capable of conferring cellular trans. In the mid-70s, and this paper was authored by Dennis Staline and Harold Varma and Peter Vogt. And what they discovered was that one of these V-oncs, the sequence was actually the same or derived from cellular sequences. And so this revealed this phenomenon of how these viruses had attained transforming capability was by transducing cellular sequences. And so they were sort of like dowsing rods, these retroviruses, for finding cellular sequences that had the potential to cause tumors. And so the one that Tony Pawson was studying in these days was GAG-FIPS from Fujinami sarcoma virus. And Tony showed that the kinase activity associated with this protein was for tyrosine residues. So, you know, a lot of kinases that phosphorylate serine and threonine, but this one phosphorylated tyrosine. He also discovered this domain. It was actually work done by Ivan Sadowski, a PhD student in the lab. And this is the famous sarcomology 2 domain. And this is really a prototypic example of a protein module that mediates protein-protein interactions. So this particular domain has the ability to bind to tyrosine phosphopeptides. And a lot of these oncoproteins were tyrosine kinases. And so this domain played an important role in allowing these molecules to talk to one another. So I've referred to it here as a typical protein-protein interaction domain, or some signal transduction glue, if you like. So FES and FIPS were discovered to be the same gene when the cellular loci were discovered. So a lot of people did the same thing that Bishop and Varmus did and found out what those proto-oncogenes were that their retroviruses encoded. And so the cellular gene for FIPS and FES looks something like this. And it encodes a protein that has a kinase and an SH2 domain, but it also has a very interesting N-terminal domain that I'll talk a little bit about later on in the seminar, called an F-bar domain. And, okay, so what F-bar domains, I'll just introduce them here. F-bar domains are only contained in FES and FER, the two of 538 protein kinases in the human genome. So that's one of the unique things about these two kinases. They have this domain. It mediates dimerization, which is important for activation, but it also mediates their ability to bind to membranes and to actually induce bending, shape changes in membranes. So this, as I say, is unique to these two kinases. So about 30 of the known approximately 100 proto-oncogenes were identified by this mechanism of retroviral transduction from the viruses that were isolated from different animals. So we know by definition, actually, that when mutated and appropriately expressed, proto-oncogenes have the ability to contribute to cellular transformation and even carcinogenesis, and that can happen in humans. So about half of these proto-oncogenes have been shown to be mutated in human cancers. But interestingly, FES is not one of them. It's never been shown, at least not so far. So I think we can all agree that these proto-oncogenes didn't evolve to cause cancer. So we're very interested in understanding what their normal functions are, in this case, FES, and understanding in these proto-oncogenes if they can contribute to carcinogenesis in any way. And finally, if that's the case, can inhibition of these proteins play any therapeutic role? So one of the first experiments that was done by Supak Yi, who is another postdoc in the lab, was to take the sequences encoding the retroviral GAG-FIPs and generate a transgene that has a promoter that will drive transcription of this gene, and introduce this into mice by microinjection of zygote stage embryos, as shown here. And what Supak Yi found was that these mice all developed tumors, and they developed a wide variety of tumors. So this was not surprising, and it confirmed that this protein is capable of causing tumorigenesis in a mammalian animal. When I started working in the lab, I got to play with human as proto-oncogene. And so what I was able to show is that overexpression of human Fez does not cause tumors in transgenic animals. And so this was the same approach. And what we found was that this particular fragment of human DNA had the whole locus, actually, the promoter and all the cis-acting regulatory elements. And so it drove what we expected, which was myeloid expression. So we knew Fez, normal Fez, was expressed in the myeloid lineage. And this just shows that we had lots of lines, lots of different copy numbers of the transgene, and expression was proportional to the copy number. So there were mice here that had 40 times more wild type Fez in them than the endogenous Fez, but they never developed tumors. We did see the expected myeloid expression, but no malignancy. And I next tried a little trick here by introducing a little additional sequence into the Fez locus that would provide a consensus sequence for N-terminal myristylation. And so now the protein gets little fatty acid, sticky finger-like motif on the end. And we thought that that would juice up its transforming capability by allowing it to bind membrane. At that time, we didn't know the F-bar could actually mediate membrane binding. So the same experiment was done, and the interesting phenotype that we saw in these animals was hypervascularity. In fact, you could tell which animals were transgenic as pups because they had what Supak Yi referred to as the blushing phenotype. And that was because they had vascularization in the subcutaneous brown adipose tissue. And images in green there are a lovely multiphoton experiment that Jeff Mewburn did for us back then that just showed the vasculature, in this case in the liver. So this was actually a fortuitous experiment because it informed us that this gene is actually expressed in the vascular endothelial cells. And unfortunately, I won't be able to talk very much about that today. So in spite of this vascular hyperplasia, we didn't see any tumors, and we didn't see any dysregulated hematopoiesis in these animals. There were subtle phenotypes, but it didn't disrupt hematopoietic lineage. It didn't result in lymphoid leukemias, lymphomas, any sort of thing. But it did show us that there was this interesting vascular expression. So when I started my lab here at Queen's, by this time I had learned some other ways of making transgenic mice besides just making a DNA construct and injecting it into zygotes. By this time, we were doing homologous recombination in embryonic stem cells. So we could target genes with very specific mutations, isolate the ES cells that had that genetic mutation, and then make a mouse out of it, get that ES cell to contribute to the germline and establish mice. And so the first two mutations that we engineered, this was work done by two very gifted PhD students, Ralph Zerngebel and Yotis Senes. And what we did was engineered a point mutation that killed the catalytic activity. So that lysine to arginine substitution at 588 kills kinase activity. Still make the protein, just has no activity. And then the deletion of exons 15 through 19 generates a null mutation. The protein isn't detectable, it's probably unstable. So we had these two models. Interestingly, we didn't see any effects on hematopoiesis again. So they don't seem to be necessary for hematopoiesis. But these mice were both hypersensitive to endotoxin challenge. And this was our first clue that this kinase may actually play a role in regulating inflammatory signaling, which ends up being very important, as you'll see. One of the things that was done with this KDR line was it was crossed into a transgenic mouse model of mammary carcinoma. And this was generated by a friend of mine, Bill Muller, who we went to graduate school together at McGill. And he made this transgene during his postdoc with Phil Leader, where he was driving an oncogenic version of the HER2 receptor in the mammary epithelial cell lineage. And what you can see in that graph on the right is that these animals, the females, all develop mammary tumors. But you'll notice that if they have either one or two alleles of FES in that mutant state, tumor onset gets delayed. So this was intriguing and suggested that there was some pro-tumorigenic role for FES. Connie Zhang, who did these crosses, observed another thing. Before tumors arose, so she was looking by immunohistochemistry at the mammary glands before there were palpable tumors. And what she noticed was that there was a much elevated frequency of T cells, as well as myeloid cells in the pre-malignant glands. And she also, by biochemical means, showed that NF-kappa B was hyperactivated. So this is an important transcription factor that drives the expression of lots of inflammatory genes. So this was intriguing because it suggested, well, maybe the reason that tumor onset is delayed is because there's this anti-tumor immunity mediated by the immune cells, both the T cells and the myeloid cells, that might be holding back the carcinogenic process. But eventually, the tumor overcomes that and tumors develop. So Sean Parsons was looking at inflammatory signaling in bone marrow-derived macrophage. And what he noticed was that if you look at NF-kappa B activation, you can activated by LPS, lipopolysaccharide, in both genotypes, wild type and null. But the magnitude of that activation is quite a bit higher in the FES null macrophage. And it also persists longer in this time course than it does in wild type. And this correlated with improved or higher levels of TNF-alpha, an important inflammatory cytokine produced by these FES null macrophages. But also what Sean noticed when he did flow cytometry and looked at the receptor for LPS, TLR4, was that whereas in a wild type macrophage, within 5 minutes, 3/4 of the receptor has been internalized. Only less than half of it has been internalized in the knockouts. And it doesn't even get down to this level after 15 minutes. And because internalization of this receptor is thought to play an important role in downregulating this inflammatory signaling, we thought, oh, that's interesting. That may be why there's persistent activation of NF-kappa B in these animals. So just to summarize to this point, we think that the delayed tumor onset in the MMTV mu mice correlates with evidence of heightened inflammation and immune involvement prior to tumor onset. The FES deficient animal macrophage here are responding to LPS to a larger extent, as indicated by NF-kappa B activation and TNF secretion. And there's also this delayed endocytosis of the receptor. So these clues suggested to us that maybe elevated inflammatory signaling in FES deficient antigen presenting cells, so that could be macrophage or more likely dendritic cells. This event during immunogenic cell death may lead to improved anti-tumor immunity. So the model that we're working on, you have to understand immunogenic cell death to appreciate this. So when cancer cells are killed by various things, in this case, radiotherapy or chemotherapy or viral infection, you will see the release of tumor-specific antigens that can be phagocytosed by antigen-presenting cells and presenting to T cells. But there's also release of these danger-associated molecular patterns that drive inflammatory signaling. And in addition to this presentation of antigen, you need these inflammatory signals to get full activation of the T cells. And then these can go and kill the cancer cells, so you start this nice loop. So our model is suggesting that FES may play some kind of a role in the third signal, the regulation of the production of these inflammatory cytokines, where the first two signals are antigen presentation through MHC1 and T cell receptor and co-stimulatory receptor being signal 2. So when Brian Light came to the lab, he wanted to study this phenomenon, so he established or did a pilot experiment using black 6 mice now. So we back-crossed our Fez targeted mice into black 6 strains so that we could do orthotopic engraftments with various black 6 derived cancer cell lines. So this E0771 is a mammary carcinoma cell line. And what you can see is that, that tumors grow slower in the Fez knockout mice. But even more interestingly, we saw that lung metastasis was a lot less in these mice. So Brian took this model and ran with it and started doing these sorts of engraftment models. And he used, in addition to the E0771 system, he added a melanoma cell line. And all of these animals are either treated with doxorubicin or DMSO vehicle to induce immunogenic cell death. And this is just the regime, the treatment regime in these models. So what you can see, I don't want to spend a lot of time going through this data, so you could believe me. If I showed you all and talked through it in 10 minutes, you'd appreciate all the statistical significance indicated. But what this data shows is that tumors grow slower in Fez deficient animals. The doxorubicin treatment is more effective in Fez deficient animals. And actually, Fez deficient animals control animals. Their tumors grow slower and survival is better than the wild type animals that are treated with doxorubicin. So this all boded well for doxorubicin treatment being more effective in Fez knockouts. To understand what was going on at the level of the immune system, Brian then started doing immunophenotyping of these tumors in addition to spleens, but I'll just show you the tumor data today. And what we were very delighted to see was that if you look at activated T cells, and so you probably can't read this, but this is CD69 as a marker of activated T cells. And then this is the CD8 population. What you see in both these models is that there's more activated T cells in the Fez knockout mice than in the wild type mice. And doxorubicin treatment actually seems to increase that frequency of activated cells. Brian also noticed that there was more PD-1 expression in the Fez knockout, especially in the melanoma model, but it could be seen after doxorubicin treatment in the mammary carcinoma model. But he also looked at macrophage, and what he saw here was very intriguing because M1 and M2 macrophage are considered to be a bit of a yin-yang model where M1 are anti-tumorigenic. They have the ability to both kill cancer cells and also contribute to activation of adaptive immunity, whereas M2 are considered to be more pro-tumorigenic, in fact, may even be immunosuppressive. So the fact that there were more of the anti-tumorigenic macrophage in the Fez knockout and fewer of the M2 boded well for these cells maybe contributing to some of this anti-tumor immunity. So it also, because of the PD-1 effect that we saw, suggested that they might be more susceptible or might be better treated with anti-PD-1. So PD-1 is one of these therapeutic antibodies that attacks the co-inhibitory signaling pathway, the checkpoint pathway in T cells. And again, these data support the idea that when you look at especially the knockout animals, you can see that the magnitude of the PD-1 effect is much, much greater. And that also translates into improved survival. So we are convinced that Fez deficiency is associated with enhanced efficacy of PD-1 treatment. So slower tumor growth in Fez deficient mice, reduced metastasis, evidence of enhanced anti-tumor immunity, evidence of improved efficacy of immune checkpoint inhibitor. These all bode well for some role for the immune system. And so we started to look more carefully at macrophage to see if they're actually contributing directly to this improved anti-tumor immunity. And to understand this, I'm going to give you a little bit of a signal transduction map here. So this is an example of what we think is going on inside the antigen presenting cell at the immune synapse. You see the T cells up here. So T cell receptor, co-stimulatory receptor, and then signal 3. So we know that Fez seems to play some role on inhibiting signaling through TLR4. When you take it out, you get more of that. And that was reflected in that NF-kappa B activation that Sean observed and increased TNF-alpha production. There was a paper published a few years after Sean's paper that showed that Fez can associate with MHC cytoplasmic domain and can phosphorylate bisphosphotase, SHP2. And they proposed that SHP2 can also play inhibitory roles on components of both of these signaling pathways. So we wanted to look more carefully at this, in addition to NF-kappa B, looking at this tank binding kinase, IRF3, its transcription factor target, as well as SHP2. And these experiments were done by culturing macrophage from bone marrow and then stimulating them in culture with LPS and then doing this sort of immunoblotting analysis. And I won't get to stare at the blots. I'm just going to show you the quantitation. These are tricky experiments that Brian and Natasha did many, many times. And what we could show was that there was indeed increased activation of NF-kappa B in the Fez knockouts, increased activation of the IRF3 transcription factor, and that correlated with increased activation of the kinase that activates this tank binding kinase. So there seems to be some intrinsic role of Fez in, we think, suppressing these signaling pathways. We also showed, but looking at RNA analysis, either RNA-seq or sorry, not RNA-seq, yeah, RNA sequencing or quantitative RT-PCR, we were able to show that a lot of the cytokines that are activated by these transcription factors are upregulated in the Fez knockout. But I'm just going to show you the data that looks at and that gets released from these cells. And what you can see here is, yes, indeed, there's increased secretion of many of these inflammatory cytokines from Fez deficient macrophage. And we're particularly interested in IL-12 because it's a key player in this signal 3. And so when we looked at the cells by flow cytometry, what we could see was that in Fez deficient macrophage, a higher percentage of them are positive for IL-12 in the knockouts. But even more intriguing, if you don't permeabilize the cell and you just look on the surface of the cell, you could see that there was more IL-12 on the surface of these Fez deficient macrophage. And if you look by immunofluorescence, so IL-12 is the yellow, this is a macrophage here. And there are a number of T cells in this co-culture that are associated with this macrophage. And it's quite striking to see that IL-12 accumulates in the immune synapse area in these cells. So this has been observed by others, and we're not quantitating a difference here in their specific accumulation in the different genotypes, but we know that that's an interesting aspect of how IL-12 may actually present to the T cells in the immune synapse in the same place where signal 1 and signal 2 are delivered. So this is sort of our model here, where not only is Fez contributing in these different ways to this upstream signaling pathway, but it might also play a role in regulating the trafficking of these cytokines out into this synaptic space. But that observation is also suggesting that maybe some, in the absence of Fez, some of this IL-12 may remain associated with the membrane and may be retained on the membrane in a form that could be presented to the IL-12 receptor on the T cells. And so Chonyan Shin, who's with us here tonight, he did these interesting experiments looking at this phenomenon in co-culture experiments. So co-culturing dendritic cells in this case with T cells and looking at activation. And actually this is work done by Brian, but Chonyan has been repeating this in dendritic cells now. Brian was doing it in bone marrow derived macrophage. So he cultures these macrophage and then you charge them up with a peptide. So they will phagocytose this peptide. And this peptide is recognized by a transgenic T cell receptor that's expressed by the CD8 T cells from this OT1 mouse. So we can just isolate splenic CD8 T cells and then add them to these co-cultures. And then after a certain length of time, recover the T cells and analyze them by flow to see if they're being activated. And what Brian and Chonyan have seen is that there is indeed increased activation of the T cells that are cultured with Fez deficient and either macrophage or dendritic cells. And that's the readout here is looking at internal interferon gamma. So interestingly, there's also more of the interferon gamma in the cells that are positive, just as determined by the mean fluorescence intensity values. So this is what these cultures look like. Sunyan gave me these pictures to thank you very much. It's quite interesting when dendritic cells are co-cultured with T cells, you can see the big dendritic cells. The T cells just seem to mind their own business. But if the dendritic cells have been loaded up with that peptide first, then because that peptide is presumably being presented on MHC1, then the T cells start to grab hold of the dendritic cells or vice versa. And you start to get these aggregates, and that can be stimulated even further by LPS, which you can see more clearly in this lower power magnification. So you can see that in the Fez deficient dendritic cells compared to the wild type dendritic cells, you get more of these aggregates with OT1 T cells sticking to the dendritic cells, and that is stimulated even further by a higher dose of LPS. So if this is the case and we're getting better IL-12 presentation to the T cells, then we should be able to see some difference in downstream activation of the IL-12 receptor in the OT1 T cells. So when ligand is presented to the IL-12 receptor on the T cells, it activates a JAK kinase system, and the substrate, one of the important substrates of that is the STAT4 transcriptional regulator. And so this is the experiment that Chanyan recently did to show that that is in fact happening. So this is wild type dendritic cells here. And you can see that this fraction in this quarter here is T cells, CD3 positive T cells that are positive for phospho-STAT4. So quantitated here is 8%. When you look at the cultures with the Fez deficient dendritic cells, you can see that there's a much higher frequency of T cells that are phospho-STAT4 positive. And just looking at the quantitation in this experiment, it's about 4.5-fold increase. So we're very excited that there's something to this role for Fez in regulating STAT4 activation by IL-12. So this is the working model that we're using at this point. We think that Fez deficiency in this antigen presentation and T cell activation paradigm enables Fez deficient cells to better activate the T cells by virtue of its effects on this signal 3 pathway, and that would lead to a higher frequency of activated and perhaps maintenance of activated T cells for longer. So what's the molecular basis of this mechanism? So I told you I'd get back to this at the end, this F-bar functionality. So this F-bar, as I said, mediates dimerization and membrane binding and bending characteristics. So what are the other proteins that have this domain? Well, there's a couple of GDBase activator proteins for members of the Rho family, but most of the other ones are adapter proteins. And what we can see in the structure of these adapters is that they have an F-bar, but they also have almost always a so-called SH3 domain, sarcomology 3 domain. So this domain is another one of these protein-protein interaction mediating modules. And in this case, there's a whole bunch of different proteins that can associate with SH3. And of particular interest in our context is NWASP and dynamin. So dynamin, some of you may know, is a protein that's involved in pinching off membranes, which is an important function when membrane or content is moving out, say, from the ER Golgi, or content is coming into the cell by endocytosis. NWASP is a protein that's involved in the formation of actin branch points on filamentous actin. And that's very important in driving membrane cytoskeletal dynamics, but also the movement of these vesicles in the cell. So this is a cartoon model of how the F-bar domain works now. So here you have this banana-shaped F-bar here. And the idea is that it can bind to membrane, but it also can bind in larger numbers and form higher order structures. And when that happens, the banana shape bends and it starts to induce that bending in the membrane. And this is not just imagination, it's been observed in cryo-electron microscopy experiments. So this is an experiment using the F-bar from one of these adapters called CYP4. And you can see here the silver is the F-bar, and it's forming this lattice work on the membrane, which you can see in green. And in higher power, you can see that this F-bar dimer here is associated with another F-bar dimer through side-by-side interactions, but there's also tip-to-tip interactions going on that appear to be important for the formation of this lattice work. Even more interestingly, if you flip this on its side, you can see the F-bar is coming into close opposition with the membrane at several places here, the membrane in green. And interestingly, these areas appear to correlate with where there are clusters of positively charged amino acids. So the idea is that those positively charged amino acids may be able to form an ionic interaction with phosphates on specific phospholipid components of the membrane. So that's the model of how F-bars may be working. And there's this really interesting early observation, you see it's over 20 years ago now, where somebody overexpressed one of these adapters, FPP17, as a GFP fusion, and saw this interesting structures in the cell. And by electron microscopy, what you could see was what were described as tunneling endosomes, as well as other things that appeared to be broken off vesicles. So this is something that we've been able to observe by video microscopy, if I can get it to work. Or not. I guess I have to turn my pen off to get that to work. Want to seem to turn off. Before. So I guess you're gonna have to take my word for it again, as they say. So what we see in these images or these movies, if I can get them to play, which I guess I can. Now I've just got a pen going around here. But hopefully what you can see, I'll turn the lights off, that'll help. You can see tunneling endosome structure proteins. So we co-expressed FPP17 and FAS with two different colors on them. And these vesicles are actually very dynamic. They're moving around in the cell. So this, we think, supports the idea that FAS can play a role in formation of these sorts of structures. And of course, it brings to the table not an SH3 domain that can pull in other proteins, but it has its kinase and SH2 domains. And so the model that we have, Wahid, you may recognize this, I believe you made this, is where kinase, by virtue of their F-bars, can dimerize and bind to membrane, but that they may associate with some other proteins that have F-bars, not FPP17, unfortunately, and that those other F-bar adapter proteins can bring in, by virtue of their SH3 domains, they can pull in things like dynamin and NWASP to induce membrane pinching or actin polymerization to drive the motility of these vesicles in the cell. We haven't been able to demonstrate this at this level at this point. It's just a working model, but we have shown, Yan Gao in the lab has shown that some of these F-bar adapter proteins are actually phosphorylated by FAS and FAR when you co-overexpress them in cells. So there may be some role for FAS or FAR in phosphorylating them to regulate their ability to recruit proteins and have their actions. So I hope I have convinced you that host deficiency enhances chemotherapy treatment in these transgenic models of triple-negative breast cancer and melanoma. We see improved tumor control in these FAS-deficient mice associated with evidence of increased anti-tumor immunity. That post-FAS deficiency seems to sensitize both the triple-negative and the melanoma model to anti-PD-1 immune checkpoint therapy. We see in these FAS-deficient antigen-presenting cells enhanced signaling through the pattern recognition receptor system. And we think that this leads to increased cytokine production and in particular surface expression of IL-12. And we think this then enables these antigen presenting cells to have enhanced T cell priming capability, in part possibly through their ability to present IL-12 or other inflammatory cytokines to activate these pathways. So what's the significance of this stuff? We think FES now, there's good evidence that it is an immune checkpoint, but it's not working in T cells the way PD-1 does. It's working in innate immune cells, and its role is to regulate inflammatory signaling. That makes it, I think, a good therapeutic target potentially to enhance the efficacy of immunotherapy, and it may also be a useful biomarker to predict which patients may be capable of responding better to immunotherapy. So with that, I'm done. 10 minutes to spare. So I'll be happy to take any questions. Or do you want to just go to the reception? David, always on David to ask a question.
So first of all, I want to give you massive congratulations for this award, and thank you for everything you've done for the department and especially for the graduate program. It's really been a brilliant thing that you've done, so thank you. I have two quick questions. One, at the end of your talk, you talked about the increased surface expression of IL-12, and then you ended up with this thing about insertions in membranes. Were you implying that that somehow helps to localize the IL-12 on the surface and present it in a more attractive way for example?
Yeah, so I think FES, I mean, this is all just hand waving at this point. These are observations, but IL-12 is there's two polypeptides in IL-12. One of them, P35, actually has an extended N-terminus that can mediate membrane association. So you think of these things getting clipped, and so that would release P35 into the lumen of the vesicle, but it doesn't always get clipped. And so Brian has an idea that maybe FES is contributing to regulation through a mechanism that regulates whether there's a proteolytic clip of that or not. So if it's not clipped, it may then just stay associated with the membrane, and when the vesicle fuses, it just stays there and it retains its partner P40. So you might have more, and there's examples of other cytokines that retain or lose their membrane tethering domains. So it may be something like that, and then how FES contributes to that, I don't know. But what we're suggesting is that FES is moving along with these vesicles by virtue of its FBAR domain. How it's doing those other things presumably has something to do with what it's impacting through its kinase domain, perhaps. So it may be regulating even a protease that's capable of doing that. Another thing that we've noticed is that there's co-localization of IL-12 receptor with IL-12. And we see that in the antigen presenting cell. So we were expecting to see IL-12 just in the T cell, the receptor just in the T cells and the ligand in the antigen presenting cells. But there's actually a lot of IL-12 receptor in the antigen presenting cells. So another idea that I am fond of is the idea that those can be traveling together and that they actually may be delivered together to the T cells. And if that was the case, then now it's not just delivering the ligand, it's delivering the receptor and the ligand to the T cells. So there are examples of signaling molecules that are delivered from cell to cell in that way. So that would be kind of cool. And I think novel, I don't know if anybody's shown that for IL-12. I have I'm open to any other suggested models that people can think of. And so are my graduate students who are working on this, including Julian here. Bruce.
So what are the prospects with FES?
Yeah, so there are small molecule active site directed inhibitors that were developed for other kinases that work against FES. And so there's a panel of kinase inhibitors that were developed for ALK, which is activated through translocation in lung cancer. And so those inhibitors actually work pretty well, like single-digit nanomolar KDs for IC50s for FES and FAIR. But they inhibit about 30 kinases to about that level. So that's the problem with active site just like proteases, they tend to be non-specific. But they, therapeutically, they're being used. And so we know that patients tolerate them, there are side effects. And that's one of the things that we've done some in vitro work. And one of the more Latin does a nice job of activating the wild type cells to have better NF-kappa B activation. But it doesn't do anything to the knockouts, presumably because there's nothing to inhibit there, and they're already activated. So that bodes. Whether I can get to a point of a clinical trial with that, or what we're also more interested in is how it can synergize. I don't think it's going to cure cancer patients. Yeah. So it may see if you get help, it may enhance that, but yeah, I think all of these things will end up being combinations. So, Sam, sorry.
I have a question regarding your so like you were environment where you have your preventive practices present. You expect the preventive expression class activity different when you start to back to the versus M1?
Yeah, so we've tried looking at Fez involvement in, say, M2 polarization, which IL-4, these sorts of experiments. We don't see any evidence that it's having a big effect. So what it's doing at that level, if you're talking about skewing, how is it skewing the differentiation M1, M2? Is that what you're asking? Yeah. We need some kind of a phenotypic readout to look. Jens looked at IL-4 stimulation of cells, and we don't see any big difference in, but the super binder, still you get predominantly serine 3 in those analysis. So we have some interesting candidates coming out of that, but the situation where we know there's lots of Fez involvement. And so that has been with dendritic cells and LPS, plus minus LPS is knockout, and just comparing the phosphoproteomics. We haven't gotten into looking at different, M1 versus M2, that sort of thing. So I think that would be tough. It would be interesting, but it would be tough to do at this point. Another immunologist has a question.
It's a very fascinating story. First of all, congratulations. You did really nice. I've never seen you.
I want to be the best person in the room today.
So I think that Greer is about the most serious scientist Chandra and me talking about. So, and it was really nice to see the journey of curiosity over the last 30 years that you have shown about that. That's all cloud in him now that you deserve. Congratulations. You've been an amazing mentor for, I think, young people, your students, and people like me who start centers and everything. So, in fact, a lot of human immunity cycles, when you think about it, a lot of action also happens in the lymph node. Have you ever looked at the lymph node and what exactly happened there when it comes to professional antigen presenting cells? You are looking for T cells out there, and you're in that as well. But anything in
I'll give you my CIHR grant to read. It has a lot of proposed experiments like that. Now that Paul Koobs is here, we're looking forward to doing intravital microscopy on the draining lymph nodes. And so Abdi's sitting on these experiments before in the context of our Ezrin work. And so we know we can see these interactions. In that case, Abdi was looking at interactions between T cells and cancer cells. We haven't looked at the antigen part of that, which of course I think is where we think Fez is playing an important role. So I'm talking to Paul about the best probably use. That subcutaneous, just like the black sick. Do it in proximity to a big node that will be amenable. We can put into these animals. And we would expect then, so that's the way it's described in the grant, have the cancer cells expressing both albumin so that the OT1 T cells will have something to chew on when those peptides are presented by DCs in the draining lymph node. So I think that's obviously a very elegant way to proceed and do that. And we're trying to learn how to do that right now. John. But I think there's only one retrovirus that causes cancer in humans, right? T cell leukemia virus. Is that your question? Why don't we get tumors caused by retroviruses?
The biology, what is the question? The feline biology. How did you map out like the process of the feline family? How many species are there? Is that telling something that might have dissect out the extended biology of the whole story?
Well, I mean, I think there's two questions here. One is why don't retroviruses in humans or various, so what is unique about the feline genome, if you like, that it is susceptible to the retroviruses can recombine with cellular genes and generate these oncoproteins. That doesn't seem to happen as much in humans, and it may be we've got some virology. I don't know if humans are just less susceptible to infection with different retroviruses. Yeah. So they're just bloody bad luck for those cats and mice. I mean, I know mice, their genomes are relatively littered with retroviral integration events compared to other species, including humans. Ours are, yeah. Sorry, John.
Oh, the highlights.
Yeah.
Just wondering if it's important, and maybe it relates to some of the questions about the viral constructs causing transformation. Seems like Fez needs to be tightly restricted to do its thing as kind of this innate immune checkpoint, but not act as an oncogene if it was overexpressed in other cell types. And just kind of wondering, like, is there any evidence that there's tight regulation of FES or things like SNPs that might alter the date of FES that kind of
Yeah, so
check?
there's not a lot of work done on transcriptional regulation of FAS, and Tom Smithgal has shown that there's methylation regulation. Sue Yi in the UK has shown that there are a couple of SNPs, one actually in the upstream furin gene, and the other, I forget which intron of FAS it is, that correlate with different transcript level. And one of those bit known about that. Ralph, as you may remember, when he cloned mouse FAS, at that point, there were sequences from humans, chickens, cat. There were a variety of species where the sequence of the promoter region was known. So he was able to see areas, oh, these are highly conserved. They're likely Sasaki regulatory elements, but there hasn't been a lot of work done on really understanding, other than the ones that I just mentioned. So whether that plays an important role in how it does, it's under control, if you like, in inflammation, I don't know. So those are wide open questions. Yeah, I think, the fact that FBAR domain, they're the only ones that have it. So I think we're more interested at this point in understanding at the molecular level, once it's there, what's it doing that's really unique. And so I think it's, like in real estate, location. So it is in a particular location, which is important for signaling. Whether it's up and down regulated, and I think maybe this gets to what Sam was talking about in different states, macrophage or other cells, we don't know that. And, every day we're opening up a different paper with single cell RNA sequencing data, and that's probably going to inform things like that. And I'm sure it's probably in the single cell RNA-seq data for tumor-associated macrophage, which has been published. There's, probably a dozen different types of macrophage that you can see in the tumor. Whether, actually, Brian has looked at some of these data sets. The problem is that the transcript numbers are not high enough to really analyze that. But I think the sensitivity of those methods are getting better. So we may find out something about differential levels of FAS expression. I think a lot of it, though, has to do more with its act. None of you guys are, what happens when you manipulate FAS in cancer cells? And I had to cut all that part out of my talk because it would have made it a lot. We're already past the hour now. We don't really see examples of it being involved in a cancer cell intrinsic way, for example. And so that was one of the things for transgenic mouse models to see myeloid leukemias, for example. We don't see that. But there are situations where it's upregulated, lactating mammary epithelial cells, FAS expression goes way up. Peter Truesdell actually showed that you can induce its expression with prolactin. I forget what else he added, but you can induce its expression. But what's that, if you remember, is it's not really upregulated at the protein level. Its activity is through the roof. And so that, I think, is a big clue to how FAS may be involved in that case in regulating secretion, because that's what those cells are doing. And of course, Ralph painstakingly measured the pup weights of animals and found that pup weight correlated with the genotype of the mother. So we think that there is a role for FAS in regulating secretion, milk production from lactating mammary gland. So it does get expressed there, but we don't see any evidence of involvement in mammary.
That'd be fantastic if you have an activation phosphor-specific antibody or something that you can kind of relate to the proximity of the protein to the membrane.
Yeah.
It's probably all happening right at that. It's really hard to study.
So do you remember our efforts to make phosphospecifics for? Yeah, it didn't work too well. Maybe we can try again. Yeah. Oh. All right, I should probably stop and let people, is here. Thank you much.